Hi everyone. In this video we're going to look at the activation of B-Raf, both the regulatory region binding to RAS, pulling away from the kinase domain, and separately phosphorylation of the activation loop, which moves it away from the P loop to turn on the kinase activity. These are segments spliced out of a longer lecture video for the biochemical techniques class. These were some new things I was trying, doing some molecular modeling in Blender, and I think some of you might be interested in seeing how this came out. The full lecture video is available on this YouTube channel if you're interested. To put this in context, this lecture video gives a review of basic protein structure, polypeptide backbone, polypeptide folding, and then it discusses different representations for protein structures, ribbon diagrams versus space fill versus surface, and kind of discusses what each of those tell you and don't tell you. And then it goes into and defines protein domains, because that's an important concept for the biochemical techniques class overall. And we use parts of B-Raf to help identify and define protein domains. And finally, we get into the details of B-Raf activation. The students examine this in their computer lab that they do. So we give them some background so they understand what they're doing in the computer lab. We show the different parts of B-Raf interacting in this activation. Most of what you're going to hear is directly from the lecture video, but I will interject some technical notes here and there for how the B-Raf model was assembled and doing the 3D animations in Blender. Previously, we only looked at the catalytic domain of B-Raf, the part that carries out its protein kinase activity. The full-length B-Raf protein is 766 amino acids in length and has 3 different domains. Here we can see a model of what the 3 domains might look like in the full-length folded tertiary structure of B-Raf. Here is the N-terminus. This is domain 1. Here is domain 2. And here's the kinase domain we looked at earlier, which is domain 3. You can see this is the same structure inside a much longer protein. This is the C-terminus. You can see how each domain is highly ordered and independently folded, and that the polypeptide between them is not folded or ordered in a consistent way. The intervening segment between the domains is loose coil. B-Raf has an exceptionally long loose coil segment in the center of the protein that is intrinsically disordered. This is functionally significant because it means that this protein can position its domains in different configurations where they can interact with each other or other proteins in the cell. This long, flexible segment is referred to as the hinge region. Here we have it stretched out so that it's easier to see the full length of it. Okay, so what are we looking at here? This model is spliced together from different crystal structures and AlphaFold models. Obviously, there are no crystal structures for the full-length B-Raf because it's way too flexible with that disordered hinge region. So to start with, the kinase domain is a crystal structure. This is PDB structure 1 UWH if you're interested. As you'll see, most of the activation loop is not modeled in this structure, and we'll talk about that more in a little bit here. The rest of it is built from different AlphaFold models. There are 1 or 2 structures that have the RAS binding domain that are crystal structures in the PDB that show the RAS binding domain, but it was easier to show the RAS binding domain together with the C1 domain here. So those 2 are together a single AlphaFold model, and then additionally separate AlphaFold models for the N-terminus and the C-terminus. You can do a single AlphaFold model for the whole thing, and it shows the intrinsically disordered hinge and kind of an odd configuration. It was easier to build this and animate it in Blender, taking these as different AlphaFold segments though. So the hinge region in the center is indeed exceptionally long, so it is to scale. It probably has some little bits of secondary structure here and there. And I looked at it. If you just do the hinge region by itself in AlphaFold, it shows some bits of secondary structure, but most of it is unordered. It's flexible, so I didn't think it mattered too much. So this hinge region is actually just modeled directly in Blender, so that's not from AlphaFold. So it is just a ribbon diagram of polypeptide that is the correct length, connecting the regulatory region to the kinase domain. And I changed the color of the ribbon diagram here because that blue is too dark. So for the rest of the video, B-Raf is shown in this reddish-brown color. Let's take a closer look at the 3 domains on our B-Raf model. Domain 3 is the kinase domain. This is the conserved catalytic part of the protein kinase enzyme. This is where the substrates bind and where the enzyme active site is. We will look more carefully at the kinase domain in the last part of the video. Domain 2 is called a C1 or diacylglycerol binding domain. Diacylglycerol is a phospholipid minus the head group, so things that bind to diacylglycerol bind to the cell membrane. Its function is to serve as a membrane anchor for a protein. To be clear, a C1 domain doesn't just bind diacylglycerol, it attaches to the phospholipid bilayer in general. It does this with a loop of nonpolar amino acids that insert into the hydrophobic core of the membrane. For pedagogical reasons, I wanted to show each domain acting independently before showing them all working together in the B-Raf activation. So here we're just seeing the C1 domain inserting into the cell membrane. So the loop that is inserting into the membrane there is the hydrophobic loop. That's the correct position on the C1 domain in B-Raf. Domain 1 binds to another signal transduction protein called RAS. Therefore, this is called a RAS binding domain, or RBD for short. RAF becomes activated by binding to RAS as part of a cell growth regulatory pathway used by most animal cells. The RAF RBD binds to a specific effector binding site on RAS. This is a crystal structure of human KRAS here. So this is a PDB accession 4EPV if you're interested. So this is a surface diagram of this crystal structure. You can't really see it from this view, but RAS gets a fatty acid acylation on lysine 182 to anchor it in the cell membrane there. So that's actually modeled into RAS, and that's how it's inserting into the membrane, even though you can't really see it from this view. I got as close as I could with the B-Raf RBD domain binding to the effector binding site on RAS. There are some crystal structures that actually have those binding together, but it's a little hard to interpret exactly in this structure how it was going to bind it. Plus, I'm binding a ribbon diagram to a surface diagram. So it's close, maybe not entirely accurate there, but I got it as close as I could. This pathway is a primary control point for tissues to stimulate cell growth. In many human cancers, mutations occur that alter the proteins in this pathway so that they become stuck in their active, signal-promoting form. Continual signaling from the RAS MAP kinase pathway leads to uncontrolled cell division. Mutations to the gene encoding B-Raf are particularly common in human melanoma, the most serious form of skin cancer. Around 60% of melanomas have a mutation at a particular position in the B-Raf gene. This mutation causes B-Raf to be stuck in its active form so that its kinase activity is always on. You will look at how this mutation affects the structure of B-Raf in your computer lab. To help you understand what you are looking at in the computer lab, let's look at how RAF's kinase activity is regulated under normal conditions in the cell. One thing to understand is that there are multiple layers of regulation that control when RAF is in its active or inactive state. This makes sense because of the importance of being able to carefully control the amount of signaling down this critical pathway that causes cells to divide. Let's look at the structural details. Here is B-Raf again, showing it with the hinge region stretched out. The N-terminal part of B-Raf that has the RBD and C1 domains is referred to as the regulatory region. And the kinase domain on the other side of the hinge is the catalytic region. RAF never actually looks like this in cells. In cells that are not dividing and that have the RAS MAP kinase pathway turned off, RAF assumes an auto-inhibited state where the hinge region is folded so that the regulatory region makes contact with a kinase domain. The hinge region looks different here so that we can animate it. Here's the regulatory region with the RBD and C1 domains, and here's the kinase domain. RAF actually needs to form a dimer to be in its catalytically active state. This can be a B-Raf homodimer or a heterodimer with another RAF family protein kinase. With the regulatory region folded across the kinase domain, this prevents dimerization and keeps RAF in its inactive state. In this auto-inhibited state, the single B-Raf molecule is free in the cytoplasm and is not bound to the cell membrane. It is complexed with a pair of scaffold proteins called 14-3-3 proteins that are not shown here. Scaffold proteins provide structural support for complexes formed by other proteins. Contact with these scaffold proteins helps to maintain RAF in its auto-inhibited state. When the cell receives a growth stimulatory signal, RAS is bound to the receptor complex and goes into its active conformation. The auto-inhibited form of RAF can then bind to RAS through its RBD domain. Here we see what this might look like in the cell. The RAF RBD domain makes contact with a specific binding site on RAS. This positions the C1 domain so that it can insert into the cell membrane. Binding of the regulatory region to RAS and the membrane forces it to release the kinase domain. The hinge can then unfold and the kinase domain can extend into the cytoplasm. The kinase domain can now form a dimer with another RAF protein. Here we are only seeing the kinase domain of the second RAF molecule, but it is also a full-length RAF that is tethered to RAS through its flexible hinge region. Okay, some technical notes on all of this. So in Blender, this was all set up as a single scene. That's the nice thing about Blender. You can set up each object, each protein, and do the animation, and then you can capture that animation from different angles. So this is all a single scene, a single animation in Blender, just moving the camera to catch different pieces of it. And this was the same scene we used to see each domain acting independently in the previous section. In Blender, you can hide different objects, you can hide different proteins to just show one thing at a time if you like. So for the long disordered hinge region, you'll see it looks different here. Trying to figure out how to animate the hinge region was the most difficult part of this project. So previously you just saw it looked like a regular ribbon diagram, and that was easy enough to do, but when you want to animate it, when you want to make it move, it gets a little more challenging. So to do that, I ended up having to build different segments of it using different pieces. So these are actually all individual objects in Blender, and then you can animate them, rotate them relative to each other. So that's how the animation is done. It's still proportionally the correct length. It just looks a little different. Also, I had some choices to make about how much of the other proteins do I want to show. I decided not to take the time to show the 14-3-3 proteins. That would be an interesting project to build that in as well. But for the students, I think it's easier just to see B-Raf by itself. I think adding the scaffold proteins would kind of get in the way of seeing what's going on with B-Raf. And then for the dimerization, as the video says, so we're not seeing the full-length B-Raf for the other partner in the dimer. We're just seeing its kinase domain coming together there. And partly that was just for time constraints, and partly to make it easier to see for the students. RAF is now poised for action. But one more thing needs to happen to turn on its catalytic activity so that it can bind ATP and phosphorylate MEK. To see what happens next, we need to zoom into the kinase domain and look at its structure more carefully. Let's introduce two structural elements that are found in the kinase domain, the P loop and the activation loop. These are conserved elements of the kinase domain, so they are found in all protein kinases. The interaction of the P loop and the activation loop is what controls whether the kinase domain is in its active or inactive conformation. Let's take a careful look at the kinase domain and identify these elements. Before we go through the rest of the lecture video, let's talk a little bit about the kinase domain. So the video will point out the P loop and the activation loop in a second here. In this crystal structure, the activation loop is unordered, it's not modeled, so it's missing. So again, I wanted to use this structure because it shows good positioning of valine 600 and phenylalanine 467. This is a hydrophobic interaction that keeps the kinase domain in its inactive conformation. So I wanted to use it for that, but it's missing the rest of the activation loop. So I actually modeled that in. So you'll see it here in a second. So to do that, I took the missing stretch of amino acids and modeled them in AlphaFold. And then, of course, it's not going to fit connecting the C-terminal end to the N-terminal end just right. So then I had to manipulate that in Blender some. And so I kind of taking that and modeled it in there. Again, this is a disordered segment of the protein. So the position AlphaFold built it in is somewhat random. If you look at different crystal structures, most of them are missing a big piece of the activation loop, but some of them show different parts of it. So in other structures, a lot of the activation loop is kind of tucked more down inside there. So the activation loop is the big loop that's on top. It's probably extending up more out of the kinase domain from between the two lobes than in reality. In reality, it's probably tucked more down inside, but still able to move back and forth. But for the purpose of being able to model it in there and show it moving for the students, this worked out pretty well. And the two phosphorylation sites you're going to see, those are spatially accurate on the structure. So those are the two residues that get phosphorylated. The space fill you're going to see for part of the activation loop and the P loop, so that was done in Molstar, just modeling in the space fill for those particular residues. And again, most of the activation loop is missing, so I couldn't do the space fill for that. I could have done it in AlphaFold using the AlphaFold model, but it would have been a little too crowded. So I decided just to use the space fill from the activation loop residues that are showing in the crystal structure. And that showed enough to show that this actually closes off the ATP binding cleft. Okay, so that's enough technical notes. Enjoy the rest of the video. You can see that the kinase domain consists of two lobes, a larger lobe on the C-terminal side and a smaller lobe on the N-terminal side. The ATP binding cleft is between these two lobes. The blue molecule shown here is not ATP, but it is an inhibitor molecule. Its binding overlaps with the ATP binding pocket, so we can use it to see more or less where ATP binds. Here is the activation loop in the C-terminal lobe, and here's the P loop in the N-terminal lobe. When the kinase is in its inactive state, the activation loop is interacting with the P loop in a way that closes off the ATP binding cleft. This structure is the kinase in its inactive state. If we look at where the electron density is for atoms in both loops, you can see that they are touching, and this blocks ATP from coming in between the loops. What sort of interaction is holding the activation loop together with the P loop? You will discover this for yourself when you do the computer lab. But this interaction keeps the kinase in its inactive state. To shift the kinase to its active conformation, two residues on the activation loop become phosphorylated. The phosphorylation disrupts the interaction of the activation loop with the P loop and pushes the activation loop more towards the C-terminal lobe. This opens up the ATP binding cleft. The activation loop movement also positions R groups in the active site so that the kinase reaction can proceed. RAF is now on, and it can phosphorylate its target substrate. This mechanism of activation of the kinase domain is conserved in all protein kinases. You can think of the activation loop as the on-off switch for the kinase. So what phosphorylates the B-Raf activation loop, you might ask? That's a good question. Remember that pulling the regulatory region away from the kinase allowed RAF to form a dimer. Most likely, the activation loop is phosphorylated by the other RAF subunit in the dimer. Although it is possible different protein kinase enzymes may also contribute to phosphorylating the activation loop. Now that you have a feel for B-Raf and how its kinase domain gets activated, you are ready to investigate some of this in more detail in the computer lab. How does the melanoma mutation that activates RAF come into play in all of this? This is what you will look at. This concludes this video, and we'll see you in the lab.